We had our last meeting in July, and with all of the COVID uproar and lots of delays and stuff, I mean, it's amazing we're still surviving today, but that's just to show how tough we are. Um, so yeah, so what I'm going to do is probably highlight the agenda for today, just to bring everybody up to speed. Um, Let me just load. Can everyone see that?
Yep.
Okay. So yeah, so I'll have a very brief introduction. Um, so, as I said, we have a very tight hour today. So this is what the agenda looks like. I'm going to kind of just reorient everyone. Take about 3 to 5 minutes to do that. But before we take off, I think I wanted to let the team know that. So we're going to get an update from Sue and some of the nice chemistry work that they're doing at CL Charles River. Then we're going to get an in vivo update from Michelle in Nada's group. So we're really looking forward to that. And I also wanted to, introduce Dr. Neil Vasdev, who, he's an expert in PET imaging. He's actually a PI and a professor at U of T. So I wanted to welcome him to the team. Um, he has some proposal. We've talked a lot about, you know, how are we going to do PD studies in vivo? I know this has been something, top of mind for Nicole and stuff. How are we going to really assess PET imaging and stuff in animal exposure, get some sort of readout for occupancy? So Neil has the appropriate background and he has expressed some interest in actually working with the open source project. And so welcome, Neil. We're going to have a 10-minute proposal from him. So we're looking forward to hearing that. And then hopefully we have enough time at the end to discuss. Um, Somebody just came in. Angela?
Yes, you
Yes.
have,
So,
not to worry, it was Diane, I believe.
okay, good. Yeah, sorry. Okay, so, but before we start off, I really want to kind of give kudos to the team. Congratulations on the GEMED Chem paper, the chemistry paper that we just published. David Smill and other members in the team really took the lead and published one of our leading medchem papers highlighting some of the open tools that we have, the likes of 2009 and 2163. So I wanted to kind of give kudos to the team for really making that happen. We also had a very special cover that was created. I think Carlos was involved in helping engineer some of that. So I think the team have done a marvelous job. And prior to that, we had another publication on some of the AMITE. So I think we're starting to get information out there for people to really dig through and utilize as they see fit in some of their research. Okay, so to begin, just to remind people of some of the actions we had from the last meeting. Some of the key things are, trying to complete some of the in vivo efficacy studies that are ongoing. And so the last time we had a nice update from Diana and Chris from ICR on the Betcher model, the orthotopic model. We were really excited to see, some of the efficacy that we saw for some of the doses in that model. We also got an update from Angle for 2163. And, we're looking forward, we're getting some additional study update from Nada Jabado and Michelle today. So we're really looking forward to that. In addition, like as I said, Charles, we will continue to kind of look at some of the, data in the database and looking at the lead compounds, trying to understand if there are ways we can further improve the potency, since if that was one of the aspects the SAB think we could actually try and improve on. So we're going to get a nice update from Sue on some of the efforts from Charles River, along with some of the efforts around the blueprint scaffold. Um, Again, just for a status update to the team again, so it's good to see that Dr. Angel have completed 2009 and 2163 in his model. Like I mentioned, Chris Jones and Deanna have completed 2009 and they're currently planning or have initiated studies on 2163. Um, and you had Michelle and Nada who was looking at 2009. So we're going to get some update on that. And the other compounding line would be 2163. So just to remind people what the two lead structures are, I just have them on this slide for the benefit of people who wanted to be reminded of what the structure of 2009 looks like. And 2163. And I just wanted to let the team know also that because of COVID, we had a little extra time granted to us by the CTIP to provide a progress update in October 15th. So that's one of the deadlines coming up. So as we get updates, we'll be responsible for putting together a progress update for the TPAC. Okay, so without any further ado, I want to first introduce, let Sue give us the update on her wonderful CRL chemistry. So let me just update, load her slides. Any questions so far? If not, so we just stop sharing here. Go to Sue's update. Sue chemistry. Yep. Okay, can everyone see that?
So can you put it to full screen, please,
Yeah,
Mifflin?
perfect.
Yep.
Yeah.
Okay, lovely. So next slide, please. Okay, so hi. So during this last period, we've spent a little bit of time making a larger sample of M4K 2163. So this was one of the compounds that Methvin's just talked about, but I think you'd requested it from other sources, but there had been some significant delays. So we offered to see what we could do here at Charles River, and I'll just there's a slide about what we've done. So our main focus on the rest of the efforts that we have available is to look at, within the main series, improving the potency, looking at those areas that give good potency, both enzyme and cell assay, but where there's been actually limited exploration. And then in the backup series, we're incorporating some of those functionalities from the main series that have provided high potency, and also looking at why is M4K 3007 so much more active than some of the other compounds, and what can we learn from that. And we're trying to make some compounds to incorporate features that do that. So obviously, we are quite limited, but RBC are continuing to screen. But we will need obviously the cell assay and some limited ADME support as and when required, when we've got some compounds of sufficient interest. So if you go on to the next slide, please. So the resynthesis of M4K 2163. So I think it was you tried to get a supply previously of this, and I think the issue that had been raised was that and we certainly found it when we started looking, is the supplies of intermediate 1 shown there with the boronic acid it was very expensive, and also it was quite difficult to get hold of, and there were long lead times. However, the bromo intermediate 2 on the right there seemed to be actually more readily available and about quarter of the cost. So we bought some of that in and thought, well, it must be quite difficult to make the boronic acid, but in actual fact it was quite easy. So we converted a significant amount of that through to intermediate 1, and that's then allowed us to make, we were targeting about 5 grams, but because of the amount of intermediate we managed to get through, we were able to make about 11 grams of the M4K 2163 HCl salt. So it is interesting to note that this material contains a very small amount of the trimethoxy compound, which is obviously 2009, and we're not entirely sure whether that actually has come, because it's only about 1%, whether that's actually come through from the starting material too, or whether at some point it's touched methanol and that fluorine has displaced. But it does contain about 1% of 2009, but hopefully that should be good enough for your requirements.
Okay.
It's actually quite difficult to remove that. We have made some efforts to try and remove it, but
okay.
so
Have you shipped it yet?
we haven't. So that was one
okay.
of the. So do you want it shipped to the OICR? To you?
Yes,
Yeah,
please, if
we'll
you don't
get
mind.
that done in the next probably next week.
Okay. Yeah, thank you very much, and thanks to the team for this heroic effort in
Yeah,
getting more material
yeah,
available.
yeah,
Really appreciate that.
yeah.
Mhm.
So we do actually have some more of that boronic acid still available as well, and the bromo.
Okay. Yeah.
So looking into the data mining on the main pyridine series, and I've been working with Lisa on this, so she contacted me after the last meeting and said, could she help? So she's been helping in terms of looking into some of the, doing a, more than just a look-see at the data. She's been digging in a bit deeper. We looked last time at some of the areas. I highlighted some of the different features that had led to some of these compounds shown here, which have got very low nanomolar activity and probably below the level that the assay can differentiate. But we can use the nanobret assay as well as a means to separate out these compounds. So we've been looking at these and what we might be able to do around some of these areas, because most of them there's been a relatively limited exploration. So particularly the 2218 and 2220, those two areas looked to provide some significant opportunities, with probably less opportunities in the 2089, but maybe one or two opportunities. So if you go on to the next slide. So looking at the first compound, 2218, the CN appears to provide an increase in potency, both in the cell and the enzyme compared to H or methyl. We also see that the chloro gives good potency. It immediately made me think about what compounds might be possible with perhaps bridged piperazines, and we found that there was only this one compound in the database which has this bridged piperazine, and it doesn't look too bad. Obviously there is a drop off, but then you may well be able to see if we can modify that. That may not be the right bridged compound. So if you, the next click please. So, so as I say, only one bridge compound's been prepared, and we would suggest that there's some further opportunities, both with the substituted piperazines and with some other bridged compounds, and there's examples here, and we can look at see whether the introduction of the chlorine or the nitrile will increase that potency. Obviously, we understand that there was some CYP activity observed with some of the nitriles, but I don't think it could conclusively be assigned to the presence of the CN on 2218. So I think there's opportunities there to make some further analogues. Next slide, please. Okay, yeah. So looking at pyrazole tails. So again, that's not 2220, the second one, sorry. A copy and paste there. Okay. So these two compounds at the top here are actually isomers. So you can see they're different pyrazole isomers, but both seem to give quite reasonable potency. And there's been relatively small amount of exploration. If you want to do the next click, please. So I thought it's worth actually looking at some of the closer match pairs. So which isomer does give us the best activity? So if you've actually got, a direct match pair, you might be able to assess which one looks best. And also looking at the compound at the bottom there, I thought was quite interesting. 2014, M4K 2014. I thought that was quite an interesting compound. It was an early compound, and it still had the 2-methyl in it. And it's surprising that's actually quite active. So I thought that there was some possibility that, well, if we take that 2-methyl, because that seemed to to reduce the activity. When we got to the 4-methyl, the 4-cyano, etc., we were seeing better activity. So I think looking at some other piperidine isomers on that pyrazole could be interesting, and also looking at an isomer with the N-linked pyrazole. I think there's another click on there as well, please. So the other thing is that this pyrazole provides a different vector. So again, the piperidine is in a slightly different orientation, and Lisa did some work on this. And she thinks that there might be the possibility by substituting on the pyrazole ring to be able to possibly improve the selectivity. So if you move to the next slide, Lisa's going to talk through that.
This is Alex from Oxford. Can I just say a couple of things? If you go back to the previous slide, we made 2 compounds taking the crizotinib pyrazole tail with one of the precursor compounds before M4K, and I can't remember whether it was similar to the 2014, something similar to that. Whether it had the methyl or not, I don't remember. We might have made both, actually. Maybe one with the NH2 and one without, with a proton, maybe. The one thing we did have with them, they got screened against the whole kinome. You know, there's a kind of reaction biology or discover it type panels on medicine. And I think we saw quite a drop off in terms of kinome-wide selectivity, more promiscuity. But I need to dig out that data and I'll share it with you all.
If you could share that, that would be useful, because
I think these are great ideas, though.
Yeah. Okay. Thanks, Alex.
Lisa.
Hi. Yes. So with regards to looking at these sort of more potent compounds, we were seeing that, for example, some of these, well, the selectivity over ALK5 was still there, definitely in the biochemical assay. It wasn't as good as compounds which weren't quite as highly potent. So I took a look at the structure of, and what I've got on, I've got a few things sort of overlaid here. I've got in purple, that's 2009. That's shown in the ALK2 structure is the grey one. I've then put over the top of that M4K 2044 as it in the alignment it has in I believe I have the structure of that as well. I don't think that's one that's in the PGB, but that was one that I've been sent from Eleanor before. And you can see that that's the structure in green. The pyrazole ring sits a lot flatter, and the amine group is also at a bit of a different angle. Now, when I've overlaid this with an ALK5 structure, I mean, there's a lot of different ALK5 structures that are available, quite exactly the same. But what's shown sort of with the orange, I haven't got the pointer actually, over the other side, other orange ones.
Yeah, this one here?
Those ones there, yeah.
Okay, yeah.
Is the sort of the twist that you have between the pyridine ring and the aryl ring. Kind of clashes with what in ALK5 is an isoleucine. We have a smaller valine in ALK2. So the green compound sitting a lot flatter, so it doesn't clash with ALK5, and you know, we see perhaps not a selective compound. So we go with this sort of idea that the flatter compound is less selective, but we then try and introduce the twist into it by some pyrazole, perhaps the selectivity can be changed. I would add a caveat to that, though, that I've looked at sort of a lot of different ALK2 compounds, and you know, this doesn't necessarily always hold. We have all sorts of sort of flatter compounds and, you know, more twisted compounds also that are selective. So, you know, but it is, I guess, one thing to investigate here and see if that can add some selectivity or improve the selectivity. The selectivity is there, it's just perhaps not quite as striking as we would like.
Okay, yeah. Thank you.
Okay, so this is one of the other probably flatter ones, looking at these tetrahydroisoquinolines, and these do show quite reasonable selectivity. And there's a couple of compounds, and both isomers actually with the nitrogen in both positions at the top, in the top compound. So nitrogen there and nitrogen up a little bit. Yeah, that one. They both show interesting activity, and it gives, we've also got the opportunity to possibly substitute on the tetrahydroisoquinoline ring in the way that we see in M4K 2218, where we've got those methyl groups on either side of the ring. So that may help us to some extent. Yeah, so as I said, in this area, I think there's less opportunity because the set of compounds are sort of H-methylchlorocyano have all been made. But you know, I think that does give us some opportunities. Again, if we do need to introduce some sort of twist to further, although these compounds have quite good selectivity, but if a further twist might be interesting, then we can always put a substituent in that ring. Yeah. And something we're doing in the blueprint series is making a compound where that's a nitrogen, where it would have the same potential interaction with tyrosine. I can't remember what tyrosine it is off the top of my head. In the blueprint series, which we see with the pyridine versions of that central ring. Okay, next slide. Okay, so moving on from the main series, going back to blueprint. So we have made a further sample of M4K 3007, which Alex had asked for. So that should have been shipped to the SGC last month, I believe. You should have received it.
Yes, I think it arrived.
Yeah. So one thing to be aware of is that the chemist who was doing this did observe some poor stability of this compound on long-term storage. So the sample that we originally had actually was quite a lot less pure than this. So it's just something to be, it's just a little be aware sort of thing, that long-term storage of this compound may not be ideal. Okay, so next slide, please. I think it's just a repeat of a slide I usually have in there, but since it's been a couple of months, I thought I'd leave it in there. So this is where we've got to really with this series. So M4K 3007 obviously is one of our most potent compounds in this series. So that has the ethoxypiperidine there. In order to reduce pKa, we changed that to the piperazine. And we do get a reduction in activity, in particular in the nanobret assay. But we do get really nice selectivity, and we get a much better PK profile. So as always with these things, it is a balance of properties. But what we're trying to see now is not only what we can do to further enhance the brain to plasma ratio, or the brain concentrations, in fact, such that we would have enough compound on board in the brain to be able to, you know, effectively get what we want, achieve the effect that we want, but also enhance the activity. So if you move on to the next slide, please. So the first compound we made was to see whether or not we needed that amide in there. So we made a morpholine compound, and it was pretty weak. So that's not the way to go. Next, please. So most of the tail modifications have shown moderate to poor activity. So if you just carry on, click. So we looked at some of these different areas of substitution. For the top area was substitution on the piperazine. So we looked at a range of different analogues. Most of them result in a significant reduction in activity. The only one that shows some moderate activity is the final one there, which was 33 nanomolar. And then if you move down into the next set, so we've got putting an additional nitrogen in there was detrimental to activity. The tetrahydroisoquinoline did show activity, but we're also looking to make the compound compound 3 where that is a nitrogen, because we do in all the other series, we have a nitrogen in that position, which does seem to enhance the activity, as I mentioned, with that interaction with the tyrosine around near the hinge. And the other recent compound that we've just got the data back recently is this amino acetidine, which also shows some interesting activity. None of these are quite good enough because I think we'll see a significant drop off in the nanobret assay. We also looked at some C-linked pyrazoles, but we didn't make the piperidine compound because the pKa of that was probably going to be detrimental to this in this series. But these two substituted pyrazoles were not particularly interesting. So move on to the next slide. So I think I showed this slide last time when we started synthesis on this. So these are the sorts of areas. So if you do the next click, so some of these are the compounds that look interesting, and we highlighted that the ethoxy on the M4K 3007 seems to enhance the activity. Excuse me. But and then at the bottom we see incorporating that nitrile, which again, as we've seen in the main series, the nitrile on the core does seem to be beneficial. So if we move on to the final slide, I think. So these are some of the compounds that we're actually making. So putting functionality either onto the piperazine or onto the central ring, sorry, onto the intervening ring, the aryl ring, to see whether we can still access the similar sort of region to those. So the synthesis of these compounds has been initiated, well, hopefully we'll get something out of them. And I think that's all. So if anyone's got any questions.
Okay. All right. Thank you. Any questions for Sue? If not.
So Nathan, I have a question for you. So with respect to trying to improve on the efficacy, on the potency, sorry, you know, I've heard a few times now about how the enzymatic assays, or the, you know, you're reaching the lower limit there, and, you know, you can look at the nanobread as well. I just wonder, is it worthwhile considering, instead of just the IC50 driving the SAR off a higher criteria, like a, you know, IC75, IC90, or, I mean, if it's always going to be proportional, it doesn't matter. But if part of the goal is to improve the potency of the compound, I know we all use IC50s, but it doesn't matter.
Yeah, that's a good suggestion. Yeah, I mean, one of the other things I was wondering too is whether or not, we can, if these compounds continue to be so potent, whether we actually change the dose range at RBC. So that's another thing. We can shift the dose range. I think we start usually at 10 and come down. So a lot of the potent compound, they just said less than. We could take a couple of our potent compounds and run a lower dose range from 1 down, and really see how much potency we can get. That's another way to see if you've really hit the bottom limit of the assay. Yeah, but those are some good suggestions. You should probably explore those. Any other questions for Sue? If not, we're running tight on time. So thank you, Sue, for that update. That was really, really instructive. Okay, so up next is Michelle. So I've stopped sharing. So
okay, I'll share my slides.
Yeah.
Okay, so you can see my slides?
Yep. Loud.
Yes.
Great. Okay, so I'm going to give today an update on the in vivo work I'm doing with the 2009 compound in sub-Q model for the DIPG. So before I start with the in vivo work, I just want to show you people that I already did a lot of work with different ALK2 inhibitors, but none of the inhibitors that are already found that we know and they are very popular, and none of them are really specific for ALK2. When I tried to use the 2009 compound, I tried it on multiple cell lines, and what's interesting is that I did a study where I tried the 2009 compound on ACVR1 knockout clones that we have in the lab. So I'm showing here two examples of the DIPG4 and the DIPG007. What is very interesting is that, as you can see in this table for the IC50 dose, you have the parental cell line, let's say, for example, the DIPG4 in this case, that responds to the 2009 compound. When you remove ACVR1 in the knockout, they become much less sensitive to the 2009 compound. The same goes for the IPG007, but what is very interesting for us for the IPG007 that we have clones in which we only remove the R206H mutation and some that we completely remove the whole ACVR1 gene. As you can see, if you still have one allele of the ALK2 of ACVR1, we still have a response that is similar to the parental, but when you completely remove the ACVR1 gene, they become much less sensitive. So this response, I haven't seen it with other compounds, and this is interesting because showing that the 2009 compound is actually specific for ALK2. And I did some assays in vitro, just to show the 2009 compound effect. And you see that I have different cell lines where I assayed proliferation for 5 days. And we do see that when I use the M4K compounds at a dose that is a little less than the IC50, of course, to keep the cells alive, but we know that the signaling is there and the effect is there, and there is a decrease in proliferation of the cells, as well as a decrease in clone formation, as shown here in the IPG36 that usually form beautiful clones. So that's interesting, and we wanted to take this compound in vivo. And for this, I tried a sub-Q model for the two cell lines, the IPG36 and the IPG4, which are both H3.1 K27M with a G328V mutation for 36 and E mutation for the IPG4. So the injection paradigm is like I injected 4 million cells sub-Q in these mice prepared in Matrigel PBS. All the mice were aged around 4 to 6 weeks. And I treated with the M4K compound 2 weeks after cell injection. I started treating. This way, like, we're sure that the matrigel is gone and the bump that is staying is tumor bump. And the treatment, I tried the 30 milligram per kilogram for 5 days on, 2 days off paradigm until the end of the study. And the end of the study means when the mouse, the tumor volume reaches 1.5 centimeter cube, and at this time we have to euthanize the mouse. So these are the rules that we have here in our animal facility. So this is where we have to euthanize the mouse. I have to note that I tried a higher dose of 60 milligram per kilogram, but it was super toxic on the mice. And really, they all died within the first week of treatment. It was really a major disaster for the mice. They lost too much weight in like a couple of days, and I have some that were just found dead, and the others I had to euthanize. This doesn't work at all in my model.
That was IP, right? That was IP dosing?
It was IP, yes.
Yeah, that might be a bit much. I mean, yeah, those doses are probably better tolerated orally, but that might be too much of a dose for them IP.
Yeah.
Okay, thank you.
Yeah. So I will first talk about the DIPG36 treatment. So I have 2 cohorts of mice that I did so far. So this is just showing the weight of the mice during the whole study, that is 13 or 14 weeks. As you can see for the vehicle, there's no really problem with the weight. Even with the M4K treated, I had 2 mice that just died at the early on, but the other that stayed are fine, even if you have a small drop, but at the end they become a bit tired. But I mean, this is 14 weeks, so it is really too much on the mice, but they are still fine. So what we found so far, so as I said, I have two cohorts. So for the cohort, the first cohort I did, as you can see for the tumor volume evolution, there is a difference in the tumor growth with the treated versus the vehicle treated mice. So as you can see in this graph, and also there is a difference in survival. So the vehicle reached the 1.5 before the 1.5 centimeter cube before the vehicle, before the M4K treated mice. In my second cohort, what happened is that I still have like the same trend of the result, but the tumor in the M4K is growing much, much slower. So as I'm showing here the images of the mice just to show that indeed we do have a tumor. It's not that we don't have a tumor, but they are growing much slower, as you can see. And already the vehicle, I sacked them all, they're all gone, but they are still, I mean, the 2009 are still ongoing. So what is really very puzzling here is the discrepancy between the two cohorts. So I should say that also, if you can see from these two, like cohort 1 and cohort 2, the tumor growth even in the vehicle started a bit later. So it could be the cells, the passage of the cells, although we still have the same trend, the same idea, the same message, but I mean, the discrepancies between the two cohorts is really like something that is not very, very nice here. So I don't know, you guys, what you think, what you have suggestions, because we're suggesting to repeat just to confirm what we're seeing. So
I agree, a repeat would be nice. I mean, as we're getting more and more accustomed to these models.
yes.
Yeah, that's not a bad suggestion. Just try a repeat and see.
Yeah, Michelle, could I just ask you, so as you point out, right, between the two cohorts, the M4K treated groups behave differently, but also the control groups, right? And likewise, if you look at the survival curves, right, in the control, the vehicle treated, in cohort 1, right, about you lose about half the mice in 38, 39 days, whereas looks like it takes probably closer to 80, 85 days in cohort 2. So given that do you have other experience from other experiments using this DIPG 36 model to have a sense of what is more typical for vehicle?
As I told you anyway, that the tumor is growing later. So in cohort 2, this is what I was pointing at. And I really think it's the passage of the cells. However, I must say that the IPG 36, when we inject in the brain, so they all grow like at the same time, like they don't, I never found a discrepancy in the growth of the IPG 36 in the brain, like latency of tumor formation. They always give tumors in 3 months. I don't know if sub-Q is I mean, they behave differently if they have different, if they are at different passages. Really, like, I don't know, it could be this.
Peter, there's something else that we have. So those, if you remember with COVID, we had to stop everything. The only thing that Michelle was allowed was to continue the injection sub-Q for the, this was almost like a miracle for the ongoing cohort. So the new cohort she restarted, we had to restart afresh a lot of cultures. So one of the things is it because those DIPGs and whatever, we maintain them as we're using them constantly. There's always something that's maintained. It's almost like they're adapted. It's just, we went back to a frozen, and maybe there is just with the adaptation took a little bit more time. But what is still very puzzling for me is that there are tumors, but they don't seem to be growing more. So it's, and so the thing that goes with maybe like just, and not a passage, but just kind of sewing a new vial, is that the vehicle only also gave tumor later, which goes with something different, but still there's no tumor in the treated one, meaning no, we haven't reached the endpoint. It's so much delayed. And so what I would like to do is stack one of those mice and see what's there in this tumor. just to kind of verify. And in any case and form, we have to do it to do more. And the idea is not only to do it on the DIPG 36, which is quite unique in that this is the only cell line that's H3.1, to my knowledge, that grows in the brain correctly. The other is really whatever we've done, and I'm sure this is the experience of others, they do not grow in the brain. This is why we started sub-Q. And she will show result for another cell line, the DIPG4, where we have barely no effect, like zero of the drug. And so we need, there's something, so I can't be making claims or anything on 1 or 2 cell lines. So we need to do more cell lines sub-Q. If the 30 milligram is the thing that we decided on, we need to go forward with, we're going to go forward with, but we need to do more than those 2 and gather on more sample. And again, any feedback from the people here, if they've done it on their own, would be quite interesting. And the other thing that we wanted to do it is, because we've generated ACVR1 knockout clone, is to do it in parallel on ACVR1 knockout clone that can still form tumor, even with a delayed latency for some, but just to see if there is also some effect. There is already a latency. It's not a complete abrogation of tumor formation, but there's a latency when you remove the ACVR1 genetically.
Okay.
Is there
I'm
any?
feeding Michelle's thunder, so Michelle should go.
Out of curiosity, is there any cell lines that normally grow slower? Yeah, go ahead.
So yes, and yes. For H3.3, there are some that grow like crazy, and others you need almost to water them. We have 2 cell lines that H3.3, K27M, ACVR1 mutant. One of them grows very fast, like you like it, you love it, and the other one is slow growing. It's there, but it's like even the tumor formation. In one, it's how much is it? It's 6 months for the one and 2-3 months for one. So it's really, they're not, even though they share the same genetic mutations for the major driver, there's something different about them. And again, this is why you always need to compare them to their own control, either their isogenic clone or to the vehicle. Otherwise, you can't be making a conclusion on
it's probably very early to make any kind of conclusions. I'm just making the note that where the cells grow slowly, it seems to be a more effective therapy. And I'm just curious if it's worth investigating that further with different cell lines, which the tumors grow slowly versus where they're more aggressive. Maybe this is points to a more useful therapy for slower growth tumors, but it's early.
I'm not sure. I really am not sure. I think it's beggars cannot be choosers. It's we can't be finding the pace. I think there is an effect on ACVR1. To what extent, I don't know. The reason why I say there is an effect is that when we genetically remove it, we have a real latency in tumor formation. So we have to find a drug that works, and the M4, the ones that you have, is the most selective. What I don't like is the high IC50. If there was a possibility of having, a drug with a lower IC50, that would, I don't know. I'm not a drug person. We're doing this. It's beyond my expertise.
Peter, Peter, any thoughts? Peter, any thoughts on what you're seeing?
Well, I think, look, these are very encouraging, right? And anytime you're moving, a model, right, that normally intracranial planted into sub-Q, right? And you're learning just as everyone else is, but there's certainly something here that is very encouraging to see. I'm not sure it's strictly, of course, this is an N of 2, right? So who knows, as Nata is saying. But I'm not sure it's strictly a proliferation rate, right? Because there are other cancer models, non-DIPG, that can grow even faster, right? And so that's not clear. One of the things I saw in your design, Michelle, the experimental design that I think is really good, the way you do it, is that, you know, you implant the tumors and then you wait for 2 weeks, right, to start dosing, because, you know, sometimes people cheat, right, and, you know, it happens a lot in the industry.
No,
So
no,
you
not in
implant
my life.
start dosing right away. Who knows what's going on?
That's
Not in
right.
my life.
So we have, I would like to make sure that all the matrix gel is gone and what is left is a tumor. So this is why I start like 2 weeks, and 2 weeks for everybody, like the vehicle, the M4K. So there are some people that wait for a certain volume for the tumor to reach, and then they start their treatment. So I never tried this paradigm. I tried just right away 2 weeks for everybody. So I don't know what's the
yeah, could you speak? And Neil has a hard stop at 12, so maybe, as I said, we only have an hour. We have about 12 more minutes.
Okay, so
Can I just ask to summarize this up though? Does anyone feel that this is not worth a repeat? I mean, we have an N of 2 with very different results, but very interesting. It strikes me that this would be something that would be fantastic to be repeated. Is there anyone who doesn't think so? And if everyone agrees, then maybe we would love it if Michelle and Nada, if you'd be in the position to repeat.
Yeah. Oh yeah, I mean, like Peter said, we are learning. So I mean, these are the new models that are evolving. We're trying to understand them. So if more we repeat them, the more confidence we build, I guess.
I think beyond repeating on the same cell line, which is important still, I want to expand it on more than the two. We have one yes, one no.
Yes.
I don't understand why the no is a no. And the DIPG4 is one tricky cell line, but it's, I would like to have, so we have other ACVR1, H3.1 and H3.3 mutant. We're going to do 2-3 more, a cohort of 5 each time, and see if, what is the trend across this. If we have consistent results, it's going to be great. Again, each time we remove the ACVR1 gene, we had a decreased growth. So that gives me some, you know, I think this is why an inhibitor may be very important.
Can I ask a quick question? The number of mice, can we increase when we repeat?
I always do 5 and 5, but you know, there are some that just, I mean, I can start with more than 5 per group, you mean?
Yeah, usually, yes. Usually we go for 10 if it is possible. I know if it is too many to handle, but if you can do 10, usually you get more statistical significance.
It's very hard. She's on her own, and it's a daily injection, and because you need to do it's not easy.
Yeah, okay.
Again,
I realize that, yeah.
This is why we're doing in batch. It's not the ideal. We're going to end up with more than 10 mice, but we're doing it in batch.
I see.
Okay. All right. Go ahead. Let's move on.
Okay. So I'll move on. So I just took the tumors that we got from the first cohort, and we just wanted to check for signaling, phospho-SMAD and ID1 gene. So this is the Western blot showing that we do have a small decrease in phospho-SMAD activity as well as ID1. And this also shows in the IHC, actually. I don't know if you can see, but so this is the vehicle phospho-SMAD, and this is in the M4K. So we do see a decrease in phospho-SMAD in the M4K. So this is Ki67, and this is K27M, just to show this is a K27M tumor. So this is promising, actually. This is interesting, and I really, this is my next step is to sack the remaining mice of the M4K, because as I told you, they're still ongoing, but we need to see what's in there too. Hopefully the effect will be more striking. So I tried the other cell lines. So Nada talked about the DIPG4, and I have only one cohort of this cell line. This is the weight change, which is also like the mice are fine all through the injection. And as you can see there, I did not have any change between M4K, the 2000 and the vehicle, tumor volume or survival. So they were really the same. Nothing have changed. And this also is reflected in the phospho-SMAD activity, and ID1. So I understand I have a low number of mice here. So I also think that the IPG4 is worth repeating, even though we didn't get any result, just to see if this result is real or there's something happened with the cells. So because we really need to confirm. So if the cell line is not responding, so it is the cell line, or it was just the experiment. So again, this is a phosphosmad IHC, which I really don't see any difference between them. So so this is actually for the in vivo part, just a couple of slides to show that I tried other compounds other than the 2009, which are the 2163, 2234, and the 2236 and 2117. And among these compounds, I'm showing here an example of the IPG7 and the IPG4. Among these, only the 2163 and the 2009 were effective. So I dropped these, and I also used them on knockout ACVR1, the same as I did on 2009. Also, it showed some specificity, although I still think that the 2009 is a bit more specific, but it shows specificity. This is for the IPG7. You can see for the IPG4, it was also even better specificity when you knock out the ACVR1. So this is another potential compound. I haven't done anything in vivo yet with it. So, but this is one potential compound that we could also use for our the IPG cell line. Just to conclude, so till now we have these cohorts of the IPG36, 2 cohorts, and the IPG4, 1 cohort. I have frozen tissue and fixed tissue, so for all these. And if there is something interesting, guys, for you that you want to do on them, so we have the material that we can provide you. So as we discussed, so we will repeat the IPG36 to confirm these changes and the discrepancies. And I mentioned to repeat the IPG4. And one option also, it would be interesting and very important, because for the model we have also, we could try to treat in orthotopic models for the IPG7. So the IPG7 does not grow sub-Q. I tried it. It doesn't grow at all sub-Q. I have another H3.3 ACVR1 mutant line with BT869. It does grow sub-Q, but it takes 9 weeks to show something. So I don't think it's an interesting model for us. So that's why these will be more interesting in orthotopic models. So this is also one option. And also, if I don't know if you have any idea if we will stick to the 30 milligram per kilogram, or we just change our injection paradigm, but we don't want to cause toxicity for the mice. So this is what I have. That's
okay. Thank you. We should follow up. I mean, yeah, 30 mg, the one that's tolerated, I'll go with that. I mean, you know, that would be the way to go. Yeah. But we should follow up in terms of like requesting, getting some of these tissues and stuff, you know, if you want to do any further analysis.
Okay.
All right. Okay. Thank you, Michelle. That's an excellent update. So we'll be in touch. Yeah, we have about 5 minutes left. So Neil, is that enough time for you?
Not really, but I'll do my best. I prepared, so Methvin had asked me to prepare a presentation for about a 10-minute talk on PET and a proposal. I'll do my best here because I have a hard stop at 12. Just to give you a heads up, this is a collaboration to take the project that you guys have and label it with carbon-11 and try and do PET imaging in the ponds. Okay, let's see what if we can get through it. As you know, PET imaging involves structure using radionuclides. So CT and MRI give you structure, PET gives you function. We have 2 cyclotrons at CAMH. One of them, basically we use short-lived radionuclides for brain imaging, carbon-11, which has a 20-minute half-life, and fluorine-18, which has a 2-hour half-life. I think most of you know that FDG, or fluorodeoxyglucose, which is a radioactive sugar, is the most commonly used PET radionuclide. It can be used to visualize in a normal individual and also in patients in cancer, baseline, and after chemotherapy. The most commonly used reaction with carbon-11 is methylation. So you can see here, you use a methylate. You can imagine methylating a phenol, a nitrogen, a sulfur. You can also do more sophisticated reactions like Stille couplings and Suzuki's as well. The most commonly used method to do a methylation was actually developed at CAMH using this methodology called the loop method, where we use an HPLC loop as the reaction vessel. So this is, I brought this up because it's relevant to the current project. For those of you who don't know us, we're the Brain Health Imaging Center. We're located just down the street from you guys at the OICR. We're at College in Spadina. We're established in 1989. We have everything to go from bench to bedside translation of radiopharmaceuticals, from chemistry through medchem, radiochemistry, preclinical work, all the way from neuroimaging, binding assays, autoradiography. We do the regulatory submissions, and we have a lot of expertise in phase 0 and phase 1 PET studies, particularly with industrial partners. We have a very large facility. We have 2 cyclotrons, 17 hot cells, many clinical scanners, and we've had over 35 PET radiopharmaceuticals developed for human use. Over 50 of those were bench to bedside at CAMH. Here's a snapshot of them. As the talk is recorded, I'll let anyone who's interested can check this out after, but you can see about half of these compounds were developed at CAMH for first in human use anywhere, and the bulk of them, the remaining compounds, were for first in human use in Canada. My laboratory spends a lot of time doing radiochemical method development. What we do is we come up with novel methods to incorporate carbon-11 or fluorine-18 into molecules. As in drug development, most drugs fail, most radiotracers also fail. So like if you think of the total synthesis principles, the goal is to synthesize any molecule. Our goal is like total radiosynthesis to synthesize virtually any molecule or radiopharmaceutical. This is challenging because most of our methods are one-step reactions with methyl iodide for carbon-11 or late-stage fluorination reactions where you bring the radionuclide in at the last step. As you know, most drugs fail, most radiotracers fail, and we have got to make the compounds, get them in vivo, see if we can do something like, for example, in this case, image the ponds. If successful, we can simplify the radiochemistry later. Just like total synthesis, the goal is to be concise. We're also trying to be concise. When we work with pharmaceutical companies, which we do extensively, and I've also had some background training in the pharmaceutical sector, we ask groups there as well as physicians within the Toronto community or in my former labs at Mass General Hospital and Harvard Medical School, we would say, what if we can label any drug for any molecule for any target? What would you be interested in? This is something that Ian Watson and I have been discussing for a long time. I was fortunate to have him join my group for a short postdoc before he went to Berkeley, and we've been thinking about this for quite some time. It's an example of a project that we did with Pfizer for a different ALK, but it's to label their 2nd generation or 3rd generation compound for anaplastic lymphoma kinase and prove that it could get into the brain so that we could visualize brain mets. They went from crizotinib to lorlatinib, which is more potent, and their rodent studies showed that it had some promise. When we look at the molecule, you see these 2 gray arrows. There's fluorine on it on a non-activated aromatic ring or a weakly activated aromatic ring using a methylamide, or we could also try and label this methyl amide, which is again also non-traditional in our field. We much prefer to label primary or secondary amines that are not these amide-like moieties. We came up with a way to do this in 2 steps using the loop method. So we hit the compound with methyl iodide. We ended up using a Bach protecting group, which is something we don't normally do, but we were able to label it. And once we had it labeled, we took it into non-human primates. We showed it got into the monkey brain and it could clear very quickly. This is exactly what Pfizer was interested in. Does the compound get in the brain and can it access brain mets? We published this in Nature Com. Our lab also has another method for fluorination where we use iodonium ilids. It's an example of fluorinating a non-activated aromatic ring. Again, this is extensively published from Nature Com, and last year we published it in Nature Protocols. But what we're trying to do is use this here with drug development. We were able to show that traditional SNAR reactions displacing a nitro group led to less than 1% radiochemical yield. When you use our iodonium ilid chemistry, we're able to get it to about 14%, which is more than enough to do human use. Here's the proposal for today. What we're looking at was obviously inspired by Methvin's paper and something we think that can get us some pilot data. Rima and I have done some grants applications together, and Ian initiated a CDA between the OICR and our labs at CAMH to try and come up with projects where we could try and do things together. We proposed to make M4K 2009. This is from discussions with Ian earlier, and then we had a follow-up discussion with Owen, Emily Murrell, who's a postdoc in our group, who's going to be leading this project, and also with Peter Ho and Ian Watson. So we discussed that we could label some of these molecules. Here's the proposal for how we could methylate M4K 2009. By looking at your structures here, we can also methylate some of your other leads. Like for example, here, we could use the precursor of 2009 could serve as the precursor for 2117. So we're trying to label all these compounds, take them into rodents and image the ponds. This is a snapshot of some of our funding and the pharma partners that we work with. And just again, thank you all. This is Emily Murrell. She's on the call. If anybody wants to follow up, please feel free to send me an email, follow up directly with Methvin or Ian or anyone else on the team, or directly with Emily. Hopefully she'll be able to take it from here. And thank you all for your attention. That's just unfortunate. I have a hard stop right now and have to jump off.
Thank you very much. Thank you very much, Neil, for joining.
Really appreciate that. All right, so that's it for today. Wayne, thanks everyone. It's a very exciting update. It's amazing how you start craving for science after a while, after being homebound for so long. But that's the way it is. So if you have any other burning questions, as I say, our is up. So thanks again, everyone. And we hope to be in touch with you soon. Okay?
Thank you all very much.
Thank you.
Cheers.
Yeah.
Thank you.
Good to see everyone.
Okay, bye-bye.
Bye-bye.